Diabetes mellitus type 2 is characterized by a combination of inadequate insulin secretion and insulin resistance. With insulin resistance, there is a decreased response to insulin by insulin-sensitive tissues, such as the liver, muscles, and adipose tissues. Genetic and environmental influences play a role in the basic mechanisms of diabetes mellitus type 2. One of the environmental influences associated with type 2 diabetes is obesity. It is the most common contributor to insulin resistance. Obesity leads to increased levels of leptin and decreased levels of adiponectin. Both of these are hormones produced by adipose tissue. The changes associated with these hormones are associated with inflammation and decreased insulin sensitivity. Increased levels of free fatty acids and intracellular deposits of triglycerides and cholesterol found with obesity interfere with intracellular insulin signaling and decrease tissue response to insulin. They also alter incretin action. This is a peptide in the GI tract that responds to food intake and increases secretion of insulin. And also, they promote inflammation. Inflammatory cytokines are released from the intra-abdominal adipocytes and induce insulin resistance and are also cytotoxic to the beta cells. Ghrelin, a peptide produced in the stomach and pancreatic islets, is decreased. This is associated with insulin resistance and increased fasting insulin levels. Obesity is also correlated with hyperinsulinemia and decreased insulin receptor density. Insulin resistance, caused by these mechanisms related to obesity, leads to decreased uptake of glucose into the cells of the organ, which then leads to increased blood glucose. Elevated blood glucose levels cause an increased demand for insulin, hyperinsulinemia, which coexists with hyperglycemia. Eventually, beta cell dysfunction develops as the beta cells become fatigued from the compensatory overproduction of insulin, or when beta cell mass is lost. In addition to the problems related to insulin resistance, factors that play a role in hyperglycemia are decreased amylin. This is the beta cell discussed in the previous slide, which is responsible for suppression of glucose. In the liver, there is an increased production of glucose. In the kidneys, there is an increased reabsorption of glucose. The alpha cells of the pancreas are less responsive to glucose inhibition, which leads to an increased glucagon secretion. And in the muscles, there is a decreased insulin sensitivity and glucose uptake, resulting in increased circulating blood glucose.